Where's my... There it is. I should know. 10 more days to go. Not including today. That's pretty crazy. I gotta do bio. I got a test and presentation on Tuesday, so we're gonna do 2 hours of studying, 2 hours of preparation, and for my presentation. That's the plan. Should I change up the music? Jazz in spring. Springtime. Let's see. I hate the spring. Too much sun. Subscribers went only. Damn. Not subscribed. I can't type in chat. That's too bad. Studying in an old library in the melancholic rain. Okay. Melancholic rain. I do love some rain. I've actually got so used to listening to rain when I sleep that without rain, it's kind of hard to sleep now. Even if it's just my fan, which kind of mimics the sound of rain a little bit, it's not enough. I need the rain. Romanticizing studying. Let's hear this. What's... Oh, shoot. What's this called? You're studying alone in an ancient library at night. Where are these ancient libraries they speak of? What a terrible picture. I cannot. I can't watch that. Actually, it's kind of good. They ruined it. What else do we got? Vintage jazz. Playing in another room, and it's raining.
And we're back in third quarter planning. Adams is quarterbacking.
You know what? I'll roll. This works for me. What do we got? This is psychedelics. This is applications. This is bullshit. New window. BCI. Brantford Collegiate Institute. The best. Molecular genetics. Oh. What is this stuff? Oh. Quite a lot. Becky Bomberry. So Facebook. Okay, so lots of stuff I should know. Nothing about this. We'll be back. Okay, I'm gonna know transcription, initiation, elongation, termination. Alright, let's see what this PowerPoint has. So, this is test 2. Okay, we'll be covering test 1. This is probably where I want to start. So, don't want this. Don't need that. I need this. Wow, that's a lot of shit. When did we do all this? Transcription. Alright. Transcription is the process in which DNA is used to make mRNA messenger RNA. RNA is single-stranded, shorter than DNA, contains ribose sugar, contains uracil instead of thymine. Okay. We'll make a transcription pod. What are these for? I don't know. It's probably useless. I need a book. There's no way. Let's use a biology textbook for a biology note. Okay, so we're in the transcription. Let's try. I'm going tree planting on Monday, which means I get to skip out on this test. This test will be Monday, and the presentations will be Tuesday. So I get to skip on the test, which means I have extra time to study. Although now I have to do a test and presentation in the same day, but that should be fine. Okay, so transcription. What is it? Process in which DNA is used to make mRNA. RNA is single-stranded, shorter than DNA, and containing ribose sugar and uracil instead of thymine. Nice. So we're looking at initiation right now. There's a lot of stuff for initiation. We can probably fill in things. Part 1. I hate this. I'm not listening to this shit anymore. It's just a bit distracting.
Introducing UFred's online BBA program. Ready to elevate your career but don't have the time.
What do you want, Mr. Cat? What are you doing over there? RNA polymerase unzips the double helix by breaking hydrogen bonds that hold the base pairs together in position. She's a crybaby. No one's crying. What? Okay, so what occurs in initiation? DNA polymerase binds at the promoter Tata sequence region of DNA. RNA polymerase unzips the double helix by breaking hydrogen bonds that hold base pairs together and positions itself. A DNA strand that has been transcribed is called a template strand. The other DNA strand is called the coding strand. Okay, now elongation. Does it really part? I swear it said part. That's weird. There should be step then. Part 1. Hmm. What do I do? Polymer sequences where the RNA polymerase enzymes with multiple copies of GPA. Note that RNA polymerase has no perforating function. Therefore, transcription is less accurate than DNA replication. Okay, what occurs in elongation? RNA polymerase starts building single-stranded RNA continuously in the 5' to 3' direction. No primer is required. As elongation proceeds, the mRNA strand separates from the DNA strand, and the DNA rewinds into the double helix. The promoter sequence is now ready to accept another RNA polymerase enzyme so that multiple copies of the gene can be made. Note that RNA polymerase has no proofreading function, therefore transcription is less accurate than DNA replication. Errors will result in ineffective proteins, but transcription occurs many times, therefore enough protein is produced to be effective. Awesome. Step 3, termination. Highlight the 3 main steps to repair the mRNA to one of these series. Termination. Damn this PowerPoint. It's a PDF. That sucks. Okay, so what occurs in termination? transcription of a gene continues until RNA polymerase reaches a termination or stop sequence. RNA polymerase stops transcription and the mRNA separates from the DNA strand. Awesome. Post-transcriptional modifications. It's a big word. Huh. It's already been 41 minutes. Wow. That's crazy. mRNA does not leave the nucleus immediately. The 5' methylated guanine cap is added to the mRNA, which protects it from digestion by nucleuses. I don't know what the ones. Fucking read it. A 3' poly A chain tail is added by poly A polymerase, also to protect it from enzyme degradation in the cytoplasm. This whole process is known as capping and tailing. These introns must be removed as they do not code for protein. Non-coding sections of DNA are also known as junk DNA. Spliceosomes remove the introns and join the exons together so that all necessary coding regions are continuous. The removed introns are broken down and nucleotides can be reused. Well, isn't that beautiful? Did you know there are 3 forms of RNA polymerase in eukaryotes? Are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Why do eukaryotes need 3 RNA polymerases? I don't know, and it doesn't tell me, but I'm sure that's okay. I might have that written down somewhere. One more thing. This is called. Okay. mRNA does not leave the nucleus immediately. A 5' methylated guanine cap is added to form mRNA, which protects it from digestion by nucleuses. A 3' poly-A tail is also added by poly-A polymerase, also to protect from enzyme degradation in the cytoplasm. This whole process is known as capping and tailing. These introns must be removed as they do not code for protein. Uncoding sections of DNA are known as junk DNA. Spliceosomes remove introns and join the exons so that all necessary coding regions are continuous. The removed introns are broken down and nucleotides can be reused. Good. Okay, so I'm done transcription. Now I'm going to do translation, and I'll probably be done with today's portion of biology review. We'll do some PowerPoint stuff or presentation stuff. I think dinner's coming up as well. What time is it? 7:21, yeah, so I'll probably have dinner soon. Might not finish all of translation before then, but I'll give it a shot. Try this. Sequence A. Determine the RNA sequence from the given DNA sequence and then the code. AATAGTAC. Whatever. Who cares? GTT. Where's GTT? Oh, that would be what? CAA. So CAA. CAA. Even GAG would be CUG or CUC. So CUC. Even if ATC would be TUG. Oh no. UAG. Okay. UAG. Doesn't exist. ATC would be UAG. Even if GAA would be CUU. Even if you are AAC would be UUG. Even if you are on AAA would be UUU. Even if you were on the GAC would be CUG. Even if you were on the right, even if you're on the right track, TGG would be AAC. Even if you're on the right track, you'll UUC. Even if you're on the right track, you'll run GAT. CU, CUA, CUA. You'll run, so even if you're on the right track, you'll run over GAG would be CUC. You run over if ATC would be UAG. Even if you run over if U GUU. Even if you just CCG, sit, GC, GCA, GCA. Okay, that's done. What a waste of time. So where'd it go? If you even if you were on the right track, you'll run over. It's get. Is there a get? What would that be? GGC. It'd be get. You'll get. It'd be UUC. 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 You'll get run over if. No. CUU. Or GUU. Run over. Get run over just sitting there. Okay, that's awesome. The cast of the ribosome in eukaryotes consists of two subunits, small and large. Role binds to the mRNA and reads it 3 base pairs at a time. The cast transfers RNA or transfer RNAs. tRNA. A small single-stranded RNA, clover leaf shape, role recognizes codons on mRNA with complementary anticodon and brings the appropriate amino acid. Each tRNA carries only one specific amino acid. Therefore, 20 tRNAs are required, but there are 64 possible combinations of nucleotide triplets. Thus, there is some redundancy, so amino acids are coded for by more than one triplet sequence. That was a period, not a comma. Wait, is that as a P? Yeah, okay. What is this? Okay, here are the steps. There are 4 nitrogenous bases. How many combinations would be possible if one nucleotide could form an AA? How many combinations would be possible if 2 nucleotides could form an AA? How many combinations would be possible if 3 nucleotides could form an AA? Whew. Okay, what is translation? A process by which a cell makes proteins using genetic information carried in mRNA. The mRNA is made by copying DNA, and the information it carries tells the cell how to link amino acids together to form proteins. That's awesome. I actually do like some aspects of DNA. I think we watched this documentary on like genetic engineering and using CRISPR and the sort of endless possibilities that you can accomplish with that. It's quite fascinating. But actually, I remember after I watched that documentary, I was like, you know what? Maybe I should look a little bit further into genetics. It's pretty interesting, and I'm not totally closed off from that idea. But ultimately, learning this stuff, I just don't find it that interesting. I enjoy how complex it is and how complex it makes human beings seem. Like that's crazy that molecular complexity, the engineering of what goes on, the machinery, the functionality. It's so fascinating. It's so in depth. It's like everything that we do in the entire world. Take the Earth as a system and condense that into a microscopic thing. And we have billions, millions or billions of those throughout our body. It's really mind blowing. Okay, what we got? Step 1 initiation. The lights don't start. That's interesting. Yep. Okay. Word initiation. Smaller rRNA subunits recognize 5' guanine cap and binds to mRNA. Small rRNA positions itself at the start codon AUG to ensure proper reading frame. tRNA with anticodon UAC and the amino acid methylamine binds to the rRNA and mRNA complex. Now the large subunit clamps onto this whole complex so that M. Why is it? It was always blocked off. I gotta look at my notes for that. Okay, I'll be back. I gotta eat dinner. Yeah, I'll be back. Honey, why you calling me so late? Cozy winter coffee shop ambience with instrumental smooth jazz music and cafe sounds. There ain't no fucking cafe. Oh, there is. It's very quiet. It's also very boring, so I'm not gonna listen to it. Cherry blossoms. Lo-fi. It's gotta be other study music genres. I'm sick of the jazz and sad ambience. Study music. What do we got? Music that makes you more inspired to study and work. Okay, let's hear this.
And welcome back to
Oh,
This
it's
Is
an
the
ad.
Office Plan.
Miller just lobbed a message way over Jones's head using.
I like the background. It's pretty cool. Nice city. This was one day ago. Oh, this is new. The creators of this lo-fi channel truly understand the needs of their audience. They've managed to capture the essence of relaxation and concentration in every track they feature. You deserve a crown. Love it. I like it. Oh, there's only 6 comments. Damn. I gotta find one of those sad things and read the comments because those things are crazy. I kinda like this though. We can listen to this for a bit. I do want to play some piano for a second. I gotta learn more piano. I just play the same thing over and over again.
Those real-time notes provide a live caption and transcript, which can be super helpful for students that may have accessibility needs. Because Otter runs
I got a like. How awesome. Shit. Okay, let's do some more work. What is this? Ah, yes, lab. When's that lab due? Sometime next week. I should probably write that down. Ugh. Okay, actually, I can take this off. It just sounds like normal lo-fi. I'm not really feeling the vibe of this one. There's gotta be some better study music out there. Relaxing music. Study music alpha waves. Okay, this is 9 years old, actually. That's crazy.
Eve Online on Mac immerses you in a universe with
No, I don't like it. Little Soul. Let's see. Jazz music of Green Gables. I actually went to the Anne of Green Gables house, sort of, where the author wrote those books. I think it was based on her surroundings, her house. Pretty nice house. Old house, pretty nice property, I should say. Very cool. They built a golf course there, which I think kind of ruins the immersion, but pretty sick. Study with me. So lecture. Looks really cool. It's a great view of the city. I don't know what city that is. What's that flag? I don't know. Maybe that could be the city, Shanghai. It's possible. Oh, this is cool looking. Let's see what this is. You know what? We'll stick with it. It's a really cool picture. It's like Gotham. Very nice. Okay. Can look you in the eyes. You look like an angel. Your skin makes me cry. You float like a feather in a beautiful world. I bit my tongue, not my tongue. I bit that too, but I bit this part of my cheek. I don't want to say my lip. I guess it's like right here. I bit it like 3 times today, pretty hard each time. But it's okay. I'm a masochist for mouth pain for some reason. I don't know why. What is this? April 15th. That would have been this week. Is that like a Monday? Wait, no, this is a long. It's really like all the way back. Okay. Here's your location. This would be. Yeah, this would. Excellent. Hello, Maxwell. How are you? I'm doing pretty good. I'm just studying for a biology. I have a test next Tuesday. I also have a presentation in biology the same day. So that'll be pretty awesome. I saw Dune 2. It was one of the best movies I've ever seen. Yeah, it's a fucking fantastic movie. Great movie. Did you end up going by yourself? Animation's done, so I'm done studying, I think, for this now. And then I'm gonna do some other bio stuff. I went by myself. The VIP theater is the best because there are no children or teenagers. That's fair. I've had so many encounters where I go to a movie theater and there's teenagers. A lot of them I probably know because a lot of teenagers go, or a lot of teenagers from my school go to movie theaters. But they're so fucking annoying. It's like they have fucking ADHD. They can't sit and watch a movie. It's like, oh, you fucking idiots. Pisses me off. Okay, where's my Phoebus Levine? Phoebus Levine. Is this it? I think this is it. Following the establishment that nucleic acids were localized in the chromosomes, early suggested that the four base molecular antibodies of thymine occur in approximately equal ratios. The Russian-American biochemist Phoebus Levine who discovered ribose sugar in 1909 and discovered deoxyribose sugar in 1929, suggested that the structure of nucleic acid was a repeating tetramer. He called the phosphate sugar basin a nucleotide. The simplicity of this structure implied that nucleic acids were too uniform to contribute to complex genetic variation. Attention thereafter focused on another component of chromosomes, protein, as the probable hereditary substance. And there's alcohol, and they bring everything you order to your seat. Oh, that's pretty cool. Even like popcorn. It costs extra though, I imagine. I wonder how much. I'm thinking it's like $10-15 more. It does, but it's not too crazy. That's good. Based upon years of using hydrolysis to break down analyzed yeast, the amino acids were composed of a series of nucleotides that each nucleotide was in turn composed of just one of four amino acid bases, sugar molecule. Where's the PowerPoint? Here it is. Phoebus Levine. Fleeing to America to escape I think it was anti-Semitism. Where's my slide? My slide's very bare bones. That's not it. I really have much to do. Like, what has she covered? References. So she's done structure. I was supposed to do these three. I think she did did she do observation and conclusions? Doesn't look like it. So I'm presenting two things. One, two, three. What is this? I should say hypothesis. I don't think it's a theory, but I think hypothesis is better. Have you read, did you read any of that book I got you? A David Goggins one? I think the acids are composed of a series of pre-nucleotides in nature. Is that the hypothesis? Maybe it is. I thought it was that. It's more to do. Well, I thought it was more to do with the simplicity of the structure. What do I have? I haven't gone to the Amazon list. Fair enough. You ever watched the show House, Max? I've been watching it a lot recently. It's a really good show. Where'd my word? Where'd my word go? Word's gone. Where's my fetus? Based upon years of work using hydrolysis to break down and analyze yeast nucleic acids, Levine proposed that nucleic acids were composed of a series of nucleotides, and that each nucleotide was in turn composed of just one of four nitrogen-containing bases, a sugar molecule and phosphate group. Okay, that's good. And then we could say the simplicity of the structure implied that nucleic acids were too uniform to contribute to complex genetic variation. There's really nothing to talk about when it comes to the experiment. Okay, so what can I add on this slide just to juice it up a little bit? Where is it here? So, go there. Okay, let's delete some of this. Experiment. Can I copy and paste this? Duplicate. No, that's not what I wanted to do. I'll just Doja. Hello, Doja. I haven't. I'm more of a Grey's Anatomy. I've never like actually watched it. I've seen clips of it, but it just looks so stupid. Like, who's the main character of Grey's Anatomy? It's that one chick, isn't it? Meredith Grey. Yeah. I don't know much about this character, but House clears. Like, House is significantly better. House is so cool. Like, he just doesn't. He's so unhinged. Says what he wants to say. You can't not love House. Where are you from? I'm from Canada, Doja. The big can. Great place to live. Okay. No, I don't want this. Go away. How do I delete you? Click on that. Copy it and paste it. There we go. We are communists. Yes. Love communism. Based upon years of using hydrolysis to break down yeast nucleic acids, Levon proposed that nucleic acids were composed of a series of nucleotides. Each nucleotide was in turn composed of just one of four nitrogen-containing bases, a sugar molecule and phosphate group. The simplicity of the structure implied that nucleic acids were too uniform to contribute to complex genetic variation. Attention thereafter focused on the other component of chromosomes, protein, as the probable hereditary substance. These are more of observance, so I'm going to put these over here. I can probably stay there. Have you ever seen House, Max? Or are you just a Grey's person? Have you been given House a chance? 'Cause I'm telling you, it's gotta be. I mean, I can't really speak because I haven't seen Grey's Anatomy, but it's gotta be better. Like, House is so elite. And isn't there like 100 seasons of Grey's? Where's my Netflix at? I gotta check this out. I swear there's like over 10 seasons, at least. House only has 8, which is a lot, and I probably won't even watch all of them ever. But it's better than 10. Let's see, Grey's Anatomy has... Oh, I don't want to watch it. Also, doesn't like every character die? 20 seasons? No way. There's 20... There's 19. What the fuck? That's insane. That's actually crazy. Have you seen it all? I've never seen House. You should give it a shot. Watch one episode. If you watch an episode of House, I'll watch an episode of Grey's Anatomy, and we can compare. I'm not even close to finishing Grey's. I still need to catch up on Star Wars. Man, Star Wars. Shit. Let me take a look at Disney. Actually, I don't know. Not sure if it will work on here. I haven't been watching any Star Wars content in a long time. The only thing that's sort of piqued my interest is Tales of the Empire. I've seen some advertising for that. It looks pretty cool. But other than that, I haven't really seen anything. The Bad Batch I should probably watch. I've seen all of season 1. Haven't finished season 2. Or have I? Maybe I finished it. I don't know. Because I think my account's shared. So it says I've watched all the episodes, but I'm not sure I actually watched them. It could have been one of my friends. So I should probably finish season 2. And then season 3. I think it's all done now. Apparently it's, I mean, I liked it. I don't know if the new season's good, but I thought the previous 2 were pretty decent. What else do I have to watch? I probably should watch Ahsoka at some point. I have no idea if it's good or not, but I haven't seen it. What else do we got? Andor. Apparently that's the best show. Should really watch that one. Currently watching Bad Batch season 3. Is it pretty good? Or is it good, at least? Star Wars Visions is actually pretty good. I enjoy watching those when those come out. Okay, that might be it. So that's not too much. Just gotta watch Andor, Ahsoka, and The Bad Batch. Only issue is, where do I have the time for that? I mean, I do watch a lot of House now, so I guess stop watching House and watch one of these shows. The issue is that I feel like I'm kind of growing out of the Star Wars, I'm just kind of growing out of Star Wars in general. I don't know. Maybe it's because it's not as good as it used to be, or I've just grown up a bit and matured, and it's just not really my thing anymore. I mean, I think there's always going to be a love for Star Wars. I don't think that'll ever go away. Like, I'll always buy whatever the latest Star Wars game is. If it looks good, I'll probably buy it. But in terms of the media, I just haven't really been watching it. I just have no interest in watching it. Although I do gotta read this book at some point. I read the first one. Pretty good book. I enjoyed it. Actually, I read it twice, the first one, because I read it when I was really young, and I read it like maybe a year ago now. It's a pretty big book. I should get into that, but I'm reading so many other things. Like, what am I reading right now? Let's see. Currently, I'm reading this book, which is a horror book. I'm reading this one, which is a sleep disorders neuroscience book. Both are pretty solid. I really like the neuroscience one. It's pretty fascinating. And after that, I'm going to read Poor Things, because it was recently adapted into a movie, and the movie was really good. It won an Oscar. So I want to read that book. And I also want to read, after the neuroscience one, I think I want to read either Astrophysics for People in a Hurry, or maybe Cal Newport, Deep Earth. This one might be a shout. Look at this. Rules for Focused Success. This would probably be a good book to read. I think it'd be useful for next year. So I should probably read that at least during the summer. So I have some new information and be more productive. That's my favorite series of Star Wars books. Astrophysics for People in a Hurry is really good. Oh, it's good. I've always been really fascinated by space. And that book I've wanted to read for a long time. I got it in Christmas, but I think it's been on my list since like 2 years ago, maybe. Not 2 years ago, maybe like a little over a year, maybe like a year and a half ago. I'm excited to read that. I've been reading a lot recently, and the issue with reading is that, at least with the books I'm reading right now, they're so much more interesting than the shit I have to learn in school right now, and it just makes me like not want to do any schoolwork and just read. It's like, I gotta do schoolwork, so I can't always read. And there's always this conflict. Like sometimes I'll read the neuroscience book in the morning, and then I'll go to my bio class, and it's just like I just completely zone out of the lesson and just think about what I just read and trying to apply it to various things, and just very deep analysis that's going on in my head. There's just so many books. Like, look at this book. This is a beefy book. This is a crazy book. The Necronomicon, H.P. Lovecraft. This is a hefty book. How many pages is this? What are we at? So this book is a cheeky, hold on. This book's a cheeky 878-page book. Not only is it a bunch of pages, there's also very small print, and every page is pretty much full. It's a very hefty book. It's also really hard to read at times because it was written in like the 1800s, or these stories were written in the 1800s or something like that. It's like an older version of English, and at some points I feel like I'm having a stroke trying to read this. That being said, there are some great stories in here. Literally, like the Lovecraftian universe. Like, what an incredible universe. Like, holy shit. Lovecraft was a fucking weird man. How do you think of this? Oh shit, I forgot. I was reading the Call of Cthulhu. I think I'm on like the last part of it. Is it the last part? I think it is. I should have finished this. I should have read this in the fucking summer. Or not the summer, in the Christmas break or something. Run out of time. I don't know. Good book. You should buy it. What else am I reading? Or what else? Other books I have on the go. We got this book on the go. This is American Prometheus. This is the book that Oppenheimer's based off of, or the movie. This is almost, I think, what is this? It's a 600-page book. Small print, like, actually, I think this has smaller print, but obviously not as big. The pages. Also a very good book. I'm only like 200 pages into this book. I'm on part 3 now. I think I haven't even got to like the bomb part. But like this shit starts like all the way back, like from when he's a child. It's really good, really enjoyable book. There's so many fucking names though. So many names. I don't remember. I literally don't even know like most fucking names. There's too many to keep track of. It's literally like years of FBI documents just composed into one book. There's a philosophy book I'm reading, or I read once in a while. I'm not too far into it, but whenever I read it, it's pretty interesting. It's not. I feel like philosophy's not a thing you can just continuously indulge in. Like whenever I read a section of a book or a new concept, you kind of had to think about it for a bit. That's how I feel, at least. That looks pretty cool. I think that's all the books that's all the books I'm reading. Well, actually, there was this book. I'm probably gonna like give this away to someone, I guess. I don't know. This is a book about dinosaurs. Now I love dinosaurs, and I thought this book would be really cool. The issue with dinosaurs is that how do you discover dinosaur facts and more about dinosaurs and develop information and theories and whatnot? Paleontology, that's how you do it. That's the field of research, right? Now, the issue with this book and other dinosaur-related books I've read is that you can't seem to disconnect the dinosaur fact from the paleontology. A lot of these stories and a lot of what this book is, which I've only read not very much of, but like every chapter starts off with a paleontology adventure, the story of people going out or to like various dig sites and digging up the bones and then drawing conclusions from that. And ultimately, I think that the boringest shit ever. Like I don't care where you found this. Okay, geographical location kind of important. You can briefly talk about that, but like I don't care about like the digging process or how hot it fucking was. Like don't spend so much time on that. I just want fucking pure dinosaur facts that are cool. And the book does have that, but there's so much of the paleontology aspect of just finding fucking bones that makes the book kind of boring in my opinion. Maybe I'll give it another shot, but too many other things for me to read right now. I actually bought this very small 100-page book to sort of compensate for that dinosaur book. This one's just 10 facts. A little bit of paleontology adventure, just sort of like a brief history on how they discovered some of these facts. This one's I don't know. I think I gave it like, I'm writing a review on it right now. It's a very short review because it's only 100 pages. But I'm going to give it like a 3 out of 5 probably. It's an okay book. It probably doesn't help my review that I read it so long ago that I don't actually remember much of the facts. But it's fine. It's fine. It's all good. Based upon you. Okay, so I have this. What else should I? I hate Canva. Canva sucks. Hydrolysis, splitting of yeast nucleic acid. Okay, so I say that, and then I gotta run some conclusions. I go to the bathroom. I'll be right back. I got a question for you, Max. Assuming you're still here, I'm not too sure. Do you believe in the theory of evolution? You're here. Okay, I don't know if you heard what I said. Do you believe in the theory of evolution? Oh, you wrote something. Mostly, I think there are some weird things that may have happened, and it may be possible that some other sentient species interfered with our development. Oh yeah, okay. So you're talking like some alien programmed us to be smarter. That's is that referring to like the doubling in size in our brain could have been some other sentient being. Because that's understandable. I mean, there's like a bunch of theories on it. Actually, the psychedelics, the Stone Ape theory, that's one of them that proposes the idea or the reason to why our brains doubled in size. I was thinking about this the other day. Evolution. It's just a theory, right? And I think most religions sort of Sort of. Or maybe we used to be much more advanced and we got massively set back by something. I see. Are you talking about like ancient Egyptian, like the pyramids and whatnot, like that sort of advanced that it's kind of seemed like we were a much more advanced civilization? I've seen like I don't know. I think I've seen on like Joe Rogan, um, these sort of like hieroglyphs or like symbolism that is found in like these ancient ruins all across the world, and the time it dates back to was sort of assumed that there wouldn't have been the technology to spread this information around. You know, we did, there wasn't this crazy transport unless we were a much more advanced civilization back then, and there was maybe some natural disaster or something that set us back. Yeah, they had some sort of knowledge that we don't know, or that we don't. It's very possible. Anyways, going back to just like the root evolution, you know, things changing over time. I feel like that theory will be proven at some point. Like assuming the current database of knowledge that we possess now, let's imagine that's a constant and in years to come we still have the data that we have today. Um, if that's the case, like surely humans will evolve different, like we'll probably evolve due to like technology, like maybe our eyes will change, we'll develop like filters to better block out like blue light emission or whatever. Maybe our postures will become worse. Like I think evolution will take place, and I think it'll be a measurable thing, which then may prove the theory of evolution. And then once that's proven, I wonder what that will do for religion. Because I'm religious, but I kind of believe that there's a creator, and I guess I call that creator God, and God may have created us, but I think it was a process. I think God is responsible for everything known in science, and all that sciencey stuff leads up to us or whatever. I don't think the mainstream story of human society's development is entirely accurate. Probably not. I mean, there's so many chunks that we probably don't know, and you know, realistically, we probably will never figure out those missing pieces. I mean, how long have humans been a thing for? Humans existed. Okay, so the early, I guess, Homo genus appeared 2,000,000, see, current evidence supports modern Homo sapiens appearing around 190,000 BCE. That's a lot of years, and I mean, I guess intelligence would have sort of developed more in that time, but at some point, I think at some point there would have been like modern humans, and like, I feel like a lot of our current groundbreaking inventions came within the last 2,000 years, and before that, everything is what, BC, right? Is that how it works? What is AE? BC, in Christianity, 80 stands right here. Oh, okay, 80 is... Maybe. We had some more advanced stuff, and then some great catalysm reset us. It's really it definitely is possible. But wouldn't there be, like, I feel like there'd be proof of that, you know? I guess it depends, right? Like if it was a massive plague-like thing, maybe that wouldn't be able to be like that could be lost to history potentially. I know, if it wiped out a bunch of people. Um, but like, I mean, depends on what they built stuff out of. Depends what they built stuff out of. Hmm. Hmm. Yeah, that's true. It's probably good enough. Good picture. They suck. These pictures suck. Some things don't stand the test of time. Do you think humans now or before had more intellectual, I guess, potential? Because what am I trying to say? Do you think humans before were smarter than current humans? Because I feel like there's like you can see a sort of a negative trend, especially in like the kids of today. But that's largely due to like uh, to the internet and cellular devices and whatnot. So I wonder if like we're actually sort of downgrading in terms of intelligence over time, or if it's because we have this blockage, which is the internet, which kind of fucks with our minds. All the stuff we build out of concrete would be gone almost without a trace in 1,000 years if we disappeared. Oh, that's interesting. Hmm. Oh, what's on my arm? I don't know what that is. The massive disaster happened, only the savage caveman type people would have been able to survive the harsh conditions. The super smart people back then probably died out. Oh. Hmm. That's an interesting theory. That's probably why we didn't rebuild at the same level of advancement. Hmm. We don't actually know. We can't say confidently that the level of advancement before us was superior to our current level of advancement. I mean, like, we got shit in space. I don't see any proof that the people before us had sent things into space. It's not impossible. I mean, I suppose. It's a big reach, though. What the fuck is this? Go away. I don't. What is this? I don't want to see. I fucking hate cameras. I literally don't know how to use this shit. Where's all my stuff? How do I zoom in? Figure 11, maybe? I have no idea what the fuck that says. Fuck Canva. I would say figure 12. Figure 12. How do I zoom in? There gotta be a way. Oh, right here. Okay. It's 100. That's very close. Why did it be such a blurry image? What if I copy this and paste it? Alright, where's the image? Oh, okay, found the picture. Just a bit blurry. Copy the link. There you go. Yeah. Me too. think I'm find a clearer picture, which sucks. Well, this one's very clear. Give me this image. Let me go here. Okay, let me try. Please work. Oh, there we go. So much clearer. Good. That's good. Oh, that's so much better. Awesome. There we go. Now I have this slide. So I'm going to take you. Place you here. Okay. Where are my conclusions at? The paper proposed that nucleic acids are composed of a series of nucleotides. think I've done enough work today on the PowerPoint. I'll have to make some finishing touches tomorrow. I'm just gonna read over my notes from today, and then I'm probably gonna end my stream. Transcription. What is transcription? Process in which DNA is used to make mRNA. RNA is single-stranded, shorter than DNA, contains ribose sugar, contains uracil instead of thymine. Step 1. Initiation. What occurs in initiation? RNA polymerase binds at the promoter TAT sequence region of DNA. RNA polymerase unzips the double helix by breaking hydrogen bonds that hold base pairs together and positions itself. A DNA strand that has been transcribed is called a template strand. The DNA strand is called a coding strand. Step 2. Elongation. What occurs in elongation? RNA polymerase starts building single-stranded RNA continuously in the 5' to 3' direction. No primer is required. As elongation proceeds, the mRNA strand separates from the DNA strand, and the DNA rewinds into a double helix. Elongation continued. The promoter sequence is now ready to accept another RNA polymerase and enzyme so that multiple copies of the gene can be made. Note that RNA polymerase has no proofreading function. Therefore, transcription is less accurate than DNA replication. Errors will result in ineffective proteins, but transcription occurs many times, therefore enough protein is produced to be effective. Step 3. Termination. What occurs in termination? Transcription of a gene continues until RNA polymerase reaches a termination or stop sequence. RNA polymerase stops transcription and the mRNA separates from the DNA strand. Post-transcriptional modifications. mRNA does not leave the nucleus immediately. A 5' methylated guanine cap is added to the mRNA, which protects it from digestion by nucleases. A 3' poly-A tail is also added by poly-A polymerase, also to protect from enzyme degradation in cytoplasm. This whole process is known as capping and tailing. These introns must be removed as they do not code for protein. Non-coding sections of DNA are known as junk DNA. Spliceosomes remove introns and join the exons so that all necessary coding regions are continuous. The removed introns are broken down and nucleotides can be reused. Awesome sauce. Translation. What is translation? A process by which a cell makes proteins using genetic information carried in mRNA. The mRNA is made by copying DNA, and the information it carries tells the cells how to link amino acids together to form proteins. Step 1. Initiation. What occurs in initiation? Small rRNA subunit recognizes 5' guanine cap and binds to mRNA. Small rRNA positions itself at the start codon, AUG, to ensure proper reading frame. tRNA with anticodon UAC and the amino acid methylene binds to the rRNA and mRNA complex, and a large subunit clamps onto this whole complex so that mRNA is sandwiched between the small and large rRNA subunits. I hate saying rRNA. Step 2. Elongation. What occurs in elongation? The next mRNA codon is exposed in the A amino site. The next tRNA carrying the required amino acid. What the fuck is that? Looks terrible. enters with its complementary anticodon and a peptide bond forms between adjacent amino acids. Note, ribosomes read mRNA in the 5' to 3' direction, adding a new amino acid for each codon read. Step 3, termination. What occurs in termination? Ribosomes eventually reaches a stop codon. The 3 stop codons are UGA and UAA. That's only 2. I'm missing one. Where's the PowerPoint? UGA and then UAG. Okay, that's what I'm missing. Okay, these 3 subcodons are UGA, UAG, and UAA. These codons do not code for an amino acid, and therefore there is no corresponding RNA. Release factor. A protein aids in the release of polypeptide chain from the chromosomes. The 2 subunits of the ribosome fall off the mRNA and translation ceases. Cool. So that's it. I think I'm done for the night. More work to do tomorrow. Probably start working a little earlier. But yeah, not too bad. I got to make some edits to make edits to these. That's on tomorrow's docket. Okay. Pretty solid. Anyways, I'm gonna end the stream. I'm gonna do the study streak update though. 227 is what that was yesterday. Today is an incredible. I didn't even erase it yet. It's going to be the incredible 220. Oh, looks like shit. 228. Wow. That's insane. Almost 230. And then we have 20 more days until 250. And then only 50 more days, or actually 49 more days, and my school will be done. So that's pretty cool to think about. Anyways, it was fun. Thanks for joining me, Max. I appreciate it. Always nice to talk to you. Have a good night, everyone on stream. I will see you tomorrow, probably. I'll see you tomorrow, maybe. We'll see. Anyways, goodbye. Have a good night.